Let's see. I think we're ready for meiosis and Mendelian genetics. Students usually like Mendelian genetics and all kinds of genetics, but we've got the rest of this unit is genetics, and then the entire next unit is also genetics. Okay, cool. Couldn't tell if that was a stretch thing or like, every once in a while I have students that kind of like, and I never really know if they're going to ask a question or not. Okay, so if you've got your notes with you, after kind of cell cycle mitosis stuff, there's this little spot on meiosis. Meiosis is pretty easy, and there's really only like 2 major ideas, or I can kind of break it into like 3 ideas that really matter that much in meiosis. And they emphasize these 3 things a lot on the AP Bio test. So anytime they talk about meiosis, they generally are asking you to like compare it and contrast it with mitosis. So rarely do they say like, explain meiosis. It'll be like, explain 2 similarities and 2 differences, or what's the main goal of this versus what's the main goal of this. It's always kind of comparing the 2. Okay, so let's say that I've got, I know you're tired of hearing it, let's say I've got a chromosome 1 from mom and a chromosome 1 from dad. Just imagine, I say this all day. You only have to hear it like once. So I've got a chromosome 1 from mom and chromosome 1 from dad. As I go through, these are homologs. As I go through the G1 phase, everything looks like this. I go through the S phase. They make sister chromatids held together at a centromere. So far, it's exactly the same if I'm doing meiosis. By the way, what's the point of meiosis? What am I trying to make that's unique in meiosis? There's mitosis and meiosis-based cell division. Do you remember hearing meiosis back in 9th grade? Okay, so if I'm doing cell division in somatic cells, so somatic cells versus gametes, which are sex cells. So I could be dividing somatic cells, or I could be dividing gametes. So what are the 2 sex cells in humans? Yeah, sperm and eggs. Kind of like ham and eggs, but not at all. That's what I always hear in my head is like green eggs and ham, ham and eggs. Somebody's like, give me some ham and eggs, and it's like sperm and eggs. So I don't do as well at censoring myself. The things that come into my head, I just sort of say them now, kind of like old men do. They just sort of like, you know, how's it going? Let me tell you about my knee. So somatic cells are non-sex cells. So this is anything that's not a sperm or an egg. So if it's liver, brain, lungs, whatever, those are somatic cells. And when they do the cell cycle, so they'll do a cell cycle, and their cell cycle involves mitosis. So they do G1, S, G2, mitosis, cytokinesis. Gametes, if I'm making sperm or eggs, see now you'll never hear it differently. Their cell cycle involves meiosis. Yes, there are 3 vowels right in a row there. I don't know why. So in cell cycle, I still do a G1 and S, a G2, and then I do meiosis and cytokinesis. So if I'm looking at somatic cells, I do something like this. I have a chromosome 1 from dad, a chromosome 1 from mom. By the time I get through an S phase, I know I'm kind of skipping steps, but by the time I get to an S phase, I'm finished. I'm hoping to have identical copies like this. So that's what I'm hoping will happen. Obviously, I'm skipping many steps in there. I'm showing what it looks like, kind of like back in, say, G1 before I copy stuff. Here, we did an S phase. Now I'm just kind of like showing you kind of out of nowhere metaphase of mitosis. And then here's like cytokinesis once we're done. Okay, so all of that is a normal cell cycle. The goal is I start with one cell, and then I'm wanting to make an exact copy. So my goal over here is copies. Goal is like identical. Identical in every single way. So when I split, I want to have the identical. I want each chromosome to be identical to its partner, and I want each cell to be identical, like everything identical. That's the goal. With gametes, it's different. I've got kind of two major goals. I want genetic diversity, which is the opposite of identical. Diversity is the opposite of identical. And then number two, I want haploid cells. My goal, if I'm doing meiosis, is in that cell cycle using meiosis, is I want genetic diversity and I want haploid cells. Over here, just to kind of clarify, these are identical diploid cells. What does diploid mean and what does haploid mean again? Diploid. Yeah, two. So what would haploid look like? Yeah, instead of having like pairs of chromosome 1s, I'd have a chromosome 1. So if you think about it, if I'm making sperm and eggs, here's the sperm, here's the egg. So one night, as I always say, mom and dad love each other very much. Mom's egg has 23 chromosomes. Dad's sperm has 23 chromosomes. Oops, that kid's going to be messed up. Hang on. There we go. Do a little genetic engineering there. So when that sperm meets that egg, they combine. So the first cell is actually like the sperm enters the egg. And so like this cell membrane is that cell membrane. The first cell is actually, the membrane is like from mom's egg. Now I've got a set of 23 from dad and a set of 23 from mom. So which of these three cells are diploid? The first, second, or third? The third one's diploid because it has pairs of chromosomes. So this is diploid. They abbreviate diploid 2N. Why? I don't know. Why not? So diploid. That means I have pairs of the right number of chromosomes. So N just means the correct number. For example, apes, they have 24 sets of chromosomes. So they should be 2N, and their N would be like 24. As a human, I'm supposed to have 23 chromosomes. I'm supposed to have a pair of 23. So what are these called? They're not diploid. They are haploid. So we represent haploid as 1N. Why is it logical that sperm and eggs should be 1N? Yeah, they both contribute. If I'm supposed to have pairs of chromosomes, then I got one set from dad and one set from mom. So dad's sperm better only have one set of chromosomes, and mom's egg better only have one. That's why I'm 50-50, right? So over here, whenever I'm doing meiosis, I want haploid cells. I want half as much DNA. Otherwise, if dad's cell was diploid and mom's was diploid, then like the kid would be quadruploid. Does that kid turn out okay? Now remember, we saw just having an extra chromosome 21 was Down syndrome. Having just a single extra chromosome 13 was like Cyclops with a horn growing out of your head. So now imagine that you had like twice as much of all of them. So that's not okay. So whenever we're doing a cell cycle with meiosis, we want haploid cells eventually, and we want genetic diversity. Okay, so I start off exactly the same. Have a chromosome 1, you'll notice I'm putting them side by side, but genetically they're the same. Have a chromosome 1 from dad, chromosome 1 from mom. I'll go through an S phase. After the S phase, I look like this. Okay, now, if I start off that way, how am I going to get like half as much DNA into these cells up here? Like here in a second, if I split, Doesn't it look like that guy's going to get 2 blue ones and that guy's going to get 2 red ones? But how much do I want each cell to have? Only 1. So what would I do? Yeah, I split again. I split into 4. Right? If over here, Grandpa starts with $20 and he wants to hand $10 to each kid, he splits it. If instead, Grandpa wants to hand $5 to each of 4 kids, then he splits and splits again. So what I'm eventually going to do so that I have half as much DNA as this side is I'll just split again. So I go through 2 rounds of meiosis. And they just name the rounds. So the first time I'm dividing, they call it like prophase 1, metaphase 1, anaphase 1, telophase 1. When I split again, they call it prophase 2, metaphase 2, anaphase 2, telophase 2. So in meiosis, in order to get half as much DNA, I just split again. So that seems pretty logical. Now, for genetic diversity, there's 2 ways we get genetic diversity. And this is where it seems to get kind of weird if you ask me. This seems risky. Early on in prophase, I know that the way I have them drawn up here, they're lined up like metaphase, but in a picture that you can't see, in prophase, these chromosome 1s and these chromosomes line up side by side each other, and they start breaking off and trading their DNA. That seems risky, but that's what they do. So they'll line up side by side. So my chromosome 1s will be side by side in prophase 1. My chromosome 2s might be over here and my 3s over here. And then in metaphase, we'll line them all up in the middle, but my 2 chromosome 1s will join next to each other, and they'll start breaking off and trading DNA. They call it crossing over. At least the name makes sense. So now, part of this blue one will break off. I'll make this a little bit bigger for the online crew. There you go. Sisters joined at a centromere. Centromere, joining 2 sisters. So part of that blue one will break off. Part of that red one will break off, and they'll trade. As long as they break off in the exact same spot and trade properly, everything's cool. If not, you're messed up. So then, that was crossing over. That gave me some genetic diversity. So now, when this guy splits over here, he's like, this one is all blue. This one is mostly blue with a little bit of red. This guy over here is mostly red with a little bit of blue. Try to make that a little bit darker for you guys at home. Success. Okay, so now I just split again. I got one kind of pure blue chromosome, and then I've got one over here that's mostly blue with a little bit of red. Over here I got one that's pure red. And then it's red with a little bit of blue. Like that. So in the end, if this is a dude, all 4 of these are sperm. Are any of these sperm genetically identical to each other? No. So did we get some genetic diversity? How did we get genetic diversity? From crossing over. So, why would I want genetic diversity in sperm and eggs?
That you don't end up with a bunch of mistakes.
Yeah. What if I ate that kid? Now I got 20 just like him. From an evolutionary point of view, I know that our evolution unit hasn't come up yet, but from an evolutionary point of view, basically, let's say you have a fish. I mean, you've seen the Nemo movie. So if I'm a fish, I might lay a few hundred eggs, and then some dude comes along and just squirts sperm all over him. I mean, you're in a pond, you can't really control anything. Sort of weird if you think about it. Like when salmon are going up to spawn, and you think of what that really means. Kind of strange. If you ever see salmon swimming around, get out of the water, because it's gross. So let's say that you got a few hundred eggs, and then some dude just comes along and just see, I stopped before I used any sound effects. So he fertilizes those eggs. You've got hundreds of fish. Now, if they're all identical and they're all susceptible to some sort of disease, aren't all your offspring screwed? Yeah. But if they're all slightly different, isn't it possible that some of them are a little bit smaller than average, so maybe they can like hide in a crack? Maybe some are bigger than average, so they can fight better or something like that. Hopefully somebody will make it. So as long as there's genetic diversity, then nature can decide which one is best. For example, and this is true, if you ever take an immunology class, I never got to. We didn't have immunology. But I've read stuff on it. And in immunology textbooks, they estimate that for any given disease, like picture the flu or COVID-19 or HIV, for any given disease, they always gave examples of viruses. A small percentage of the population is immune to it. So let me give you an example, speaking of sperm and eggs. So they go to some village somewhere. I don't remember where it was, but it's kind of a third world country, so they don't have access to like condoms and safe sex type stuff. So all sex is unsafe sex, right? They just kind of like, well, it works out kind of thing. And so they're in this particular community, and the HIV rate's really high. Let's say that it was like 20-25% of females were HIV positive, and males were similar. So they're like, all right, this is crazy. I don't currently have any students named Tommy, so I'll say Tommy. They're like, Tommy over there, doesn't he like sleep with everybody? Like, isn't he sort of a man whore? And they're like, yeah, totally. How's Tommy not HIV positive? Tommy, how many partners have you had? He's like, this week? 6, and 3 of them were prostitutes. And you're like, which ones? Those 3. And they're like, all of them are HIV positive, and then 2 of those other ladies are. How many women have you slept with? He's like, 50. Which ones? Well, I forgot, but here's like a couple dozen of them. And they're like, man, a third of the women he sleeps with are HIV positive. How's he not HIV positive? Yeah, some of y'all are like, I've got friends like that. And so, how's he not HIV positive? Well, then they go and they find out he has a mutation. He's immune to HIV. They go on, check it out even more. I want to say something like 6% of some place in Europe is immune to HIV. In order for HIV to work, we spend a unit on the immune system a little bit later. And again, don't quote me on that 6%, but it wasn't like 0.0001. It was like 4% or 6% or 7%. It wasn't like less than 1%. So you don't have to know this at this moment. But one of the ways that you get sick from HIV is you have helper T cells, and they have a protein on them called CD4. You have another protein called CCR5. And for helper T cells to do their job, they need CD4 and CCR5. Well, it turns out HIV, the reason that HIV doesn't infect my skin cells, but it infects helper T cells, is that HIV grabs on to CCR5 and then pulls itself inside. So this red thing is HIV. So the way that HIV gets inside of my cells is it grabs onto CCR5 and pulls itself in. Well, it turns out this dude has a slightly mutated version of CCR5. It's still good enough to where his immune system works, but it's different enough that HIV can't grab on. So he can't get HIV. Now he's got chlamydia, all versions of hepatitis, syphilis, gonorrhea. He has every like milky white pus-like discharge and burning sensation and pustule that there is, but he doesn't have the HIV. Because by random chance, his CCR5 was mutated. Why is it mutated? Because somewhere along the way, these genes got shuffled. So if you live in a community where, pretend that's hundreds of years ago and we didn't know how anything worked and HIV was a thing, if you live in a community where like a third of the people are HIV positive, then how do you survive? CCR5 mutations. It's thought that like this year when the flu hits, that a small percentage of the population, even without a vaccine, is just naturally immune. It's highly likely that some of us have a slightly tweaked version of ACE2 receptors. That's what spike proteins in SARS-CoV-2, COVID-19 attached to. So some of us have slightly different versions of that. So we're actually immune to COVID-19, and we don't even know it. So it might just be like, hey, I didn't get the flu this year. I just assumed I was lucky, but it could be that I was actually immune to the flu of 2018 whenever it came around. So we want genetic diversity because then hopefully somebody can survive. Okay, there's one other way to get genetic diversity. So we'll say like, I'll put like, since genetic diversity is number 1, I'll say crossing over is like 1A. You kind of see my logic? The second one is harder to explain with writing. So hopefully they don't ask you to explain this in writing on the AP Bio test. I can explain it with markers pretty easily, but if I was like going to try to write it in paragraph form, it's tricky. Okay, so in meiosis, chromosome 1 from dad, chromosome 1 from mom. By the time I'm done with an S phase, I have copies. If I was going to do mitosis, they line up head over heels and separate. But in meiosis, they don't. They line up side by side by side by side. In your book, they might even call this a tetrad. Tetra means 4. So in meiosis, you have tetrads. In mitosis, you don't because they line up head over heels. So if you come across that, they line up side by side by side by side. 1, 2, 3, 4, tetrad. While they line up, they do some crossing over. Probably wouldn't need it on the AP Bio test. I think I have it in your notes. But the process of them coming together is synapsis. You technically could have these guys come together in synapsis and not do crossing over. In theory, I could take these guys, glue them together, they trade, but then they trade right back, and nothing happened. I kind of make the comparison like this. If I take a piece of yellow paper and I glue it to white paper, I could call that synapsis. Whenever I pull them apart, isn't it likely that some of the yellow and some of the white switched with each other? So it's likely that crossing over happened. Technically, is it possible that I pull them apart and all the yellow stays here and all the white stays here? So synapsis is bringing the chromosomes together to form a tetrad. And pretty much always, they're going to trade DNA, which means crossing over will happen. The crossing over didn't have to happen. They could come together and not cross over. Okay, so that's happening up here with my chromosome 1s. So let's say they do some crossing over. So I have genetic diversity. There you go. Now, here's my chromosome 1s. Here's the second type of genetic diversity. It's called, let's see if yeah, that shows up okay. It's called independent assortment. I don't want that to say 2A, and I don't want it to be an orange. So this is 1B. It's genetic diversity, and it's the second thing that causes it. And it is called independent assortment. Independent assortment. So that's probably kind of hard to read because I'm writing sort of small, but that just says independent assortment. So I've got 2 ways of getting genetic diversity. I've got version 1A and 1B. So crossing over happens, but there's also independent assortment. So here is independent assortment. These guys cross over, let's say. Hang on, everybody's all crossed over now. All right, so these guys did some crossing over. Those are my chromosome 1s. But also, I've got chromosome 2s. And my chromosome 2s lined up side by side by side by side. And they also did some crossing over. See if I can manage to not drop these. I'm not going to risk it. I'm not living on the edge. So here's my chromosome 1s. As it is right now, when you look at this set of markers that's going to go that way, that's going to go to your right, when I'm eventually 1, 2, 3, 4 cells, isn't that far cell over there going to get the red with the blue cap and then the pure orange one? That's what they're eventually going to get? Okay. What if in this cell they were floating like this, though? Wouldn't that far right cell get the red one with the blue cap and then the black one? So chromosome 1s sort independently from chromosome 2's. Independent assortment. Chromosome 1's don't determine how chromosome 2's line up. It's not like, oh, because blue's over here, then like the orange one's behind it or the black one's behind it or something like that. So 1's and 2's sort independently. When my chromosome 3's are in the middle, they could have lined up like this or this. They might be like this. So whatever happens with chromosome 1 doesn't predict what will happen with chromosome 2. It's not like because I got the orange marker here, I have to get the blue one down here. If I get the orange one up there, I could have gotten this one, or this one, or that one. Chromosome 1's sort independently from chromosome 2's, which sort independently from 3, which sort independently from 4. So what that means is before you know it, there's like hundreds of thousands of combinations, right? I guess it'd be like, crap, even without crossing over, wouldn't that be like 4 to the 23rd power? Just from independent assortment, and then I'm doing crossing over. So there's like almost infinite possibilities. So that gives me more genetic variety. Because of independent assortment, that's why I can't tell how intelligent you're going to be based off of your hair color. Because chromosome 1's have nothing to do with chromosome 7's, and maybe the IQ genes are on chromosome 7, and your hair color is on chromosome 1. Otherwise, recruiting for AP Bio would be easy. It'd be like, how dark's your hair? And you're like, it's blonde. I'd be like, get out of here. You know, your hair's not dark enough. On a scale of 1 to 10, you need to be a 7 or more. If you're tall, that has nothing to do with your intelligence. I can't tell based off of whether you're a male or a female, whether you're going to like biology. So most genes sort independently. We're going to learn in the next unit, though, that that's not always true. Sometimes they stick together. For example, let's say that you have really, really dark brown or black hair. What are the odds that I can guess your eye color? I bet about 95% correct. So how many of y'all would say that you have, like on a scale of 1 to 10, your natural hair color, on a scale of 1 to 10, we'll just call it like an 8 or darker. Like mine's probably an 8 or a 9 or something. So how many of y'all would say that you're an 8, 9, or 10? Okay? Does anybody with their hands up have something other than brown eyes? Got 1. So I'd be right. There might have been 10 of you guys, so I'd have been right 9 out of 10 times. Random chance would say 50-50 on dark eyes and light eyes. So generally, hair color, eye color, and skin color are linked. They don't sort independently. So sometimes we can tell something about you. Sometimes we can look and say, oh, you have a widow's peak. That's a dominant gene, where your hairline comes to a point as opposed to being straight across. That's actually genetic. It's one of the few things that we think is still caused by a single gene. So we might be able to say, like, oh, you have a widow's peak, and your earlobes are unattached. Mine are unattached. I have an actual lobe. Some of you guys, your earlobes just join right to your head. You never noticed it. You never noticed it, but that means you're a mutant. Yes?
Well, what about people who have like maybe 2 different eye colors or 2 different hair colors?
Yeah, so heterochromia gets weird. Basically, eye color is more complicated than this, but for the moment, I'm going to pretend that it's simple. So if you have a gene, let me pull up a picture really quick. We're getting ready to get into Mendelian genetics, so we're like basically done with meiosis. Mendelian genetics is next in your notes. So to answer your question about eye color, so if we look at eye color, we're all supposed to have brown eyes. Have I told you this before? Have I or not? Okay, so with Mendelian genetics, and I'll let you know when we're kind of in the part, like the notes part where you'd need to worry about writing something down. Right now, just try to sort of kind of worry more on understanding and less about what would I write down. We're supposed to have brown eyes. There's no such thing as a blue-eye gene. The blue-eye gene is just a broken brown-eye gene. So the way that eye color works is eye colors, all of them, whether it's blue, green, brown, whatever, are all caused by how much dark pigment you have in your eyes, how much melanin you have. So melanin is what makes my hair dark, my skin dark, and then my eyes dark. So I have genes for melanin. I know that because I have dark hair and dark eyes, so I'm not albino. If I don't have genes for melanin, if the genes for melanin are broken, then I can't put any dark pigment in my hair, my eyes, or my skin, and I'm albino. So for me, you're like, wait a second, if you have pigment for melanin and your hair is dark and your eyes are dark, why isn't your skin darker? Because the genes that say I have genes for melanin, and then I have genes that say put it in my eyes, and those genes are on. They work. They're not broken. Then I have genes that say put melanin in my hair. They're on. The genes that say put it in my skin are mostly muted, so my skin is kind of pale. I still have melanin. It's just the genes that say put it in my skin don't work much until I go out in the sun. Then those genes turn on, and now I get a tan. So if you have blue eyes, this does not explain green eyes. We're just explaining brown versus blue for the moment. Then raise your hand if you have blue eyes. Like if your eyes are largely considered blue and not green. So I see a couple. Two. Do you have blue eyes, Cade? Okay. So we'll go ahead. Because of the blonde hair, I'm going to go with blue. So we got 3. You 3 all share the same common ancestor from about 9,000 years ago. The same great-great-great-great-great-great-grandma or grandpa. They've still not overturned this. We might overturn this in the future, realize that we found that blue eyes evolved separately, but as it stands in science at this moment, we think blue eyes have evolved once. So about 8 to 10,000 years ago, I'm sorry, 6 to 10,000 years ago, so I'll just call it 8,000. 6 to 10,000 years ago, we think is the original mutation that caused blue eyes. They're like, well, how do you know that brown eyes are right and blue eyes are wrong? Because in my DNA, this is me. I have brown eyes, unlike you freaks with blue. Just kidding, it's jealousy. Everybody knows. If you have brown eyes, like I have no idea where I am on like the 1 to 10 scale, the standard 1 to 10 scale. But I know if I had blue eyes, I'd be at least 1 or 1.5 higher. Right? Uh-huh.
But if blue eyes are a recessive gene, then how did we end up with them this far down the line if only one person?
Think about that. You have a sordid family history.
But even the first person would have had to reproduce with someone with a different eye color, right?
Unless it was their own kid.
But they had
All blue-eyed people are born of incest. The question was, yeah, suck it, blue-eyed people. Back at you. All us brown-eyed people are just like, what color are your eyes? Brown. And yours like, mine are blue. Just messing with you, of course. But yes, so we think that it went back to a single mutation. I don't know if it's a man or a woman because we don't know. What that means is like, I've got the gene sequence for brown. And this sequence says, make melanin and put it in my eyes. So some of you guys, though, like right here, you know the sequence of like A's and T's and G's and C's in the middle of DNA. If you have blue eyes, your sequence is identical, except for a small piece. So along in here, all of a sudden, there's one little spot where you have like a mutation or two. And so because of that, you don't put any melanin in your eyes. So it's almost like, pretend that you have a cake, and we'll come back to incest. So let's say you have a cake, and let's say that step 3 in baking a cake is like put some brown food coloring. I will come back to I know that we kind of got off topic on the original eye color question, so I think it was Isabella. So we'll come back to that. And so let's say that step 3 for making a cake is I'm supposed to put brown food coloring in the frosting. And then I don't. I mess up that thing. What color is the frosting? It's just white. Same thing with blue eyes. Blue eyes is actually a lack of pigment. You think it's blue, but it's not. It's clear. When light bounces around and refracts, that's what I get for talking smack to you blue-eyed people, and bounces around and comes back, it appears blue, but it's not. just like the sky. You're like, the sky is blue, and you're like, but the sky is made out of the same stuff between you and I. It's clear, but if I see a lot of it on the horizon, it bends and it looks blue. And then when the sun sets and the light bends even a different way, all of a sudden it looks like pink. But the sky is clear, right? So when I look at the ocean, I'm out in the middle of the ocean, and I'm like, man, that's bright blue. But I take my beaker, because I got one, and I dip it in the ocean, and I look at it, and I'm like, wait, the water's clear. And you're like, no, it's not. It's blue. Same thing with your eyes. When there's no pigment in there, it bounces around and bends and, again, refracts and reflects and all that. It comes back out, and it appears blue. But it's not. It's clear. Just like how the clear sky and the clear water, when there's a bunch of it and the light's bending, it looks blue. But it's not. Only when you shine a light on it. Because when you shine a light on it, you're actually seeing their retinas in the back. Just like back in the day, you guys haven't known crappy cameras. But if you go and look at like grandma's photographs, when somebody looks right at the camera, it looks bright red. That's because you're taking a picture of their retina. Your eye is hollow, so light goes in. There's like goo in there, aqueous and vitreous humor. So like light goes in there, and it hits the back of your eye. And the back of your eye is red. So if I shine a light if you have blue eyes and I shine a light, if they're super light and I shine a light, I might see just a little bit of red come through. But if you have like albino blue, then more red comes through. But even me with brown eyes, if I look directly at a camera that was made in like 1990 and you take a picture, where my pupil is will be a red spot because you're taking a picture of my retina. Huh? Yeah, and so with modern cameras, it's like a red eye reduction was what they called it years ago. Modern cameras are, they're really good at it, so they sort of correct for it. But older cameras couldn't, and so if you ever look directly at a camera, you'd look like you have two little bitty red dots where your pupils were.
So we're like gray and purple and blue, kind of all like the same mutation, like
So the question was, what if you have something other than blue eyes? There's more than one gene. So eye color is what they call polymorphic, and it's dictated by many different genes. So real eye color gets complicated, is what I would say. So yeah, that's all I want to say right there. We eventually get into like breaking Mendel's rules, but imagine I have like 5 different genes that determine my eye color. And some of them are like dominant recessive. You remember how sometimes you had like co-dominance or incomplete dominance where you might like blend red and white make pink? Some of the genes for eye color do co-dominance too. So you can get weird crap. Like even if you have brown eyes, most people don't have pure brown. There's like little chunks of green in them or something. So, or you might have blue eyes with a little sunburst thing around the middle. So wish I had that. Okay. So I don't even remember what your original question was, Isabella. Oh, you're asking about like heterochromy. I don't know what causes heterochromy where you have like Siberian Huskies have it a lot where you have like a brown eye and a blue eye. So I don't know about that, but you also had a second question. And we started talking about eye color and incest so much I forgot what it was. Okay. So with hair color, like there's only melanin that you put in there. So when you have more or less, then that's what makes like black versus brown versus blonde. So blonde is kind of just more like dilute brown if you want to think of it that way. I don't know if that was your question.
Well, like my question, because like my hair, for example, because I'm mixed, so like the top of my head is like really dark brown, but the bottom of my hair is just naturally lighter. So is the melanin just different?
So that's right. That's what your question was. Sorry, that was my fault. I'll switch cameras so it feels like I'm talking to you too, a little bit more. So when you have different colors, like for example, I have gray hair now. So if you were to take my hair follicles and look at them, the ones that make the gray hair, I still have the genes for melanin, but they're just off. So I don't put any melanin in there. Now, why it is that genes that insert pigment on like maybe hair up here is like all black, and then right here it's like a little bit more brown or something like that, I'm not sure. But you do have the genes for black if your hair is black. It just, if you don't turn them on as much, then your hair would look brown. And if you turn them on even less, it's blonde. And hair color does change sometimes whenever hormone swings are big. So puberty or when women go through menopause, then hair like color and texture can change. Eye color very, very rarely changes after like 6 months of age. So people think their eye color changes but their shirt changed. So when I'm wearing a green shirt, my eyes look different. That's the whole idea of makeup, right? You look a certain way, then all of a sudden you put some like purple crap here, and your eyes look big or small or whatever. So your eyes didn't really change. It's an illusion. So I don't know, kind of like the pond out in front of Timber Creek. It looks blue, and then when the sun sets and the sky's pink, all of a sudden it looks pink. It's not pink. It's an illusion. It's a reflection. So your eyes generally don't change color that much, but your hair can. So I got one more over here, and then I got something in the chat there. So I won't forget. I've got a couple in the chat, so I will get those. Yeah. Sorry. Blue eyes, and I have brown eyes. Let's say my kids have brown eyes. So does blue eye gene please help? So blue eyes is recessive if we're talking about just brown and blue. So, for example, my dad has blue eyes. So that means he's little b, little b. That's the only option. Brown's dominant. My mom has brown eyes. So she's big B and an I don't know what. She might be another big B, and that's why she has brown eyes, or she might be big B, little b, and that's why she has brown eyes. I don't know. So I can have brown eyes. I can have brown eyes, and then here I don't know. So two blue-eyed people, it'd be very genetically, it'd take a lot of genetic trickery for two blue-eyed people to have a brown-eyed kid. But like my wife and I have brown eyes. We can still have a blue-eyed kid. I know that I'm a carrier for blue eyes because my dad has blue, so I have to have a little b from him. I know because I have brown eyes, I had to get a capital B from my mom. So I know that I'm this, big B, little b. If this is my wife, I know that she has brown eyes. I have no idea if she's a carrier. Then we could still have kids with blue eyes. So even though brown's dominant and we both have brown, we could still have a blue-eyed kid. So I don't need to like go searching or something like, where you been? Okay, so I've got a few in the chat. Let's see what we got in the chat. Is it the same for asymptomatic people? So back to, this is probably related to the COVID thing or the HIV thing. I get 2 copies of a gene. So I get one from mom and one from dad. So I could get a good copy from mom and a bad one from dad. Kind of like I've got one good copy of brown eyes, and then I've been punking on all you blue-eyed people for a while, but I carry the blue-eyed gene too. So I'm also the product of incest. The reason that if I have blue eyes, or if you have blue eyes, you have to be the product of incest is if it only occurred once, we think. So now it just occurs in this one random person. They get the blue-eyed version of the gene. They won't even have blue eyes, but they'll be a carrier for it. So let's say it mutates once. They have some kids. That kid's big B, little b. That kid's big B, little b. How does the first blue-eyed person come about? Well, there's mom, son, and son. So mom and son, that's the first blue-eyed kid. Eight 8,000 years ago. If it really only mutated once. Now, again, we might find out tomorrow, oh, remember that thing we used to say about incest? That was wrong. We were just being disgusting. So yeah, if you're blue eyes and it did mutate once, the reason when we think it mutated once is because the blue-eyed gene is the same. Like, it's the same. We can find the same little defect in DNA. Okay, let's see. We got another one in the chat. What about for hazel eyes? Because 3 separate color skin, how does that work? So I'm going to avoid the like hazel versus green for now, but there are polygenic traits and there are polymorphic versions of traits. So essentially for now, all I want to say about like hazel eyes or green eyes is there's more than one gene that determines eye color. So I know that's not a very satisfactory answer, but if you flip forward just a little bit in your notes, you don't have to. I'll just show you. We do address it some with polygenic traits right there. We'll actually address eye color more in the future. So it's a good question, but I don't want to talk about it yet. You need a little bit more genetics background before we can. All right. Pretty interesting stuff. So speaking of genetics, how about that Gregor Mendel? Pretty exciting monk, wouldn't you say? So Gregor Mendel was a monk. I've never been a monk, But I think, like, you get up and you pray, and after that, like, your day's just kind of wide open. You know? Like, I don't think you work at Subway. So, Gregor Mendel's a monk, so he basically starts doing experiments in genetics. It turns out that, like, Darwin and a lot of the other people back in the day would sometimes, like, join the priesthood or join clergy and stuff like that, because I'm not saying that, like, being a pastor of a modern church or something's easy, but sort of back in the day, like, if I lived in a commune or whatever, I don't have a lot of stuff, but, like, I don't have a lot of stuff to maintain either. So, like, I got to get up and I got to eat, and then I guess later on I'll eat again and kind of, like, tend into the garden or whatever. So Mendel started doing experiments. And here's what he found. Let's say, and this does get interesting, by the way. You're like, man, we were talking about all this cool stuff, and then you bring up Mendel. It's good, I promise. So Mendel noticed something. Let's say that I had some red flowers and some red flowers, and I crossed them. What color, before you knew anything about genetics, what color would you be expecting the offspring flowers to be if I cross red flowers with white flowers? Yeah, I'd expect pink. That's what happens with the color wheel or whatever. So if I've got red flowers and white flowers, pink flowers. So up until Gregor Mendel was like late 1800s. Up until Gregor Mendel, what would happen is scientists would do this. They'd be like, all right, here's some red flowers. Let's cross them with white ones. What color do they make? Red. That's weird. All right, and then they just moved on. Nobody had an explanation for it. So when Gregor Mendel was around, all we knew was that you kind of looked like your mom and you kind of looked like your dad. At the time, they thought that a sperm contained a teeny tiny little person that was curled up in there. So they'd have drawings, and they'd look like this. There's a sperm. And there's you. Might be a boy, might be a girl. So dad provides the sperm, and then that sperm just divides and divides and divides and becomes a baby boy or a baby girl. And I've said it before, but this seems ridiculous, but it doesn't seem as ridiculous as like DNA. DNA seems a lot more weird than that. So Mendel noticed something. He's like, all right, I'm going to take some red flowers, and I'm going to cross red with red with red with red. They're actually purple, but I'm picking red and white because we know what happens when I mix those. Like what color we expect. We expect pink. So I have these red flowers. I breed them with red, with red, with red, with red, with red. So now, whatever causes redness, right? DNA hadn't been discovered yet. Whatever causes redness, I think this one's pure red. And then he would take flowers that are white. He'd breed white with white with white with white, and he's like, all right, whatever causes them to be white, I think that these are just that. So these are purebred red, purebred white. These are what we'll call the parent generation. So then what he does is he crosses the red ones with the white ones. Most flowers have both male and female reproductive parts. So this dude, a monk, would break the male reproductive parts off of a flower and rub it on the female parts of this flower to force them to breed with each other. I find that ironic. You might not. It's like he's like this weird third wheel in the, never mind. So I cross red with white. And here's what he finds. 100% of them are red. And that was surprising. Again, every scientist before him couldn't explain it, so they just moved on. So 100% red. And it's full-strength red. I can't tell the difference between that red and that red. They're both like just as dark as each other. It's not diluted or anything. So that's what they call the F1 generation. You don't really have to know this, but F stands for filial, and apparently in Latin, filial means sons. So the F1 generation, we would say, your children or your grandchildren, but then if you're talking about goats, you have to say like the kids and the grandkids, and if you're talking about dogs, you have to say puppies, or if you're talking about cats, you have to say kittens, so they just say F1. So now what he does is some plant incest. They're plants, they don't care. And so this plant right here, he breeds it with its like, I guess it's siblings. So he takes F1 plants and breeds them with F1 plants. So he takes these red ones and breeds them with each other. And here's what he gets. Red and white. So let me clarify. I crossed red with white, and what did I get? 100% red. Then I crossed this red with another one of its siblings, red, and what did I get? Red and white. That's puzzling, isn't it? So then he's like, we kind of look like mom, we kind of look like dad. Well, it looks like whiteness wasn't destroyed. So when red and white are crossed, it looks like white is gone, but it's just hiding somewhere because it pops back up. So whatever codes for white isn't destroyed, it's still there. So then Mendel's like, hey, I know how this might work. Maybe mom provides something and dad provides something, and one of them is dominant. So kind of like if I have these two pens and you get both, maybe red always wins out. So if I get 2 genetic codes for red, then I'm red. If I get a code for red and blue, red wins out. So I call red dominant, and I call this one recessive, but it's still there. So he came up with the idea of dominant and recessive. And then another dude, like years later, came up with the Punnett square. His name was Punnett. And so basically Mendel sort of backward engineered this. He realized that the ratio of red to white was 3:1. In fact, his data, he did this with peas because he was trying to be dull. This is supposedly his data. Some people argue that his data is too good so that this isn't true. But what do you expect when you're looking at documents from 160 years ago? He'd look at seed shape, and he'd cross spheres with spheres, and then wrinkled with wrinkled, and cross those with each other. Cross those with each other and see what their kids look like. And he got like 5474 spherical, 1850 wrinkled. Let me switch cameras for you. He looked at seed color, 6,000 yellows versus 2,000 greens, 3:1, 3:1, 3:1, 3:1, 3:1. He's like, all right, this is not chance. There's something going on with this 3:1 thing. And so he's sort of like, what if red is dominant, and I get a version of the red, I get a version of the color gene from mom, and maybe one from dad, and then white was recessive. So when I cross this parent with this one, big R, cross with big R, little r. Got to switch back cameras again. So maybe red is big R, white is little r. So when I cross big R with little r, all of them turn out red. That's how I know that red is dominant. If all of them turned out white, then white would be dominant. So that means all of these kids are big R, little r. All right. Well, when I cross the kids with the kids, then now I get red, 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 white. Red, 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 white. That's how I got the 3:1 ratio. So that's how I figured it out. Now, to put this in perspective, and then I'm going to share something that's surprising with you. This is a little bit surprising, but the next thing is very surprising. So to put this in perspective, about 60 or 70 years later, Watson and Crick figured out that DNA was a double helix. Mendel could predict what your kids were going to look like, but they didn't know that DNA was the genetic material yet. So imagine if there was somebody that could solve a calculus problem before algebra was invented. How weird would that be? What's the derivative of this? What's the derivative of 3x to the third? Well, it's like 9x to the second power. And it's like, how do you do that? I don't know. All right, here's all these plants. What color are they going to be? About 300 will be yellow and about 100 will be purple. Then it comes out, it's like 301, and, it's like, man, that's almost perfect. How does that work? Or what causes genetics? I don't know. It's just math. That's what the math told me. I can predict all this stuff, but I can't explain it. Isn't that weird? Like, it's almost like if I had ESP and I could read your thoughts. How are you doing that? I don't know. I can't tell. I just do it. So that's kind of amazing. Here's another thing that it's amazing. You think you understand what dominant means, but you probably don't. So now, back to this. You notice anything odd about this dude? Yes. He's the world's greatest piano player. He can play chords that nobody else can play. It's also a joke. My jokes aren't very good. He's a 40% better piano player than everybody else. Ooh. 7 fingers, he has 40% more. All right. This one's likely photoshopped. I didn't get it from a reliable source, so that one's probably fake. However, this one right here is not fake. That's a baby. That's a baby's hand, to clarify. There's a thumb, so 1, 2, 3, 4, 5, 6. These are just gloved hands from, a medical provider, a nurse, a doctor, whoever's holding this baby's hand. This is a newborn. I can tell because his skin looks like newborn skin. On the feet, 1, 2, 3, 4, 5, 6. So it's called polydactyl. Poly means many, and dactyl, if you think of like pterodactyl, their wings are actually the same bone structure as our hands. It's just they have really, really long fingers. But if you start looking at the anatomy, like they have one joint right there, and then they have 2 joints here, and 2 here, and 2 here. So it's the same bone anatomy. It's just like now all of a sudden these bones extend all the way to the wall. So polydactyl, many fingers. Turns out being polydactyl is dominant. It's not recessive. Dominant does not mean most common. Dominant just means if there's a tie, 6 fingers wins out. Now, you're like, wait a second, Mr. Cotton, I understand math. If big R You're like, I understand math. If big R is dominant, then like anytime there's a tie, that should be more common. But the assumption that you're making that's wrong is that you assume there's an equal number of like big R's and little R's and big F's and little F's out in the world. There's not. So big F is 6 fingers on each hand and 6 toes on each foot. Little f is 5 fingers. Just turns out that out there in nature, it's like a 5,000 to 1 ratio. So everybody in here has little f, little f. If I throw a bunch of letters into a container, just little cards, and I throw 5,001 cards in there, one of them has a big F, and then 5,000 of them have little f's, and then in the gene pool of life, you come up and grab 2, one from mom and one from dad. You grab 2 of them out there, don't you pretty much get little f, little f? And how many fingers do you have on each hand? 5. What if you get the one big F and a little f? How many fingers will you have on each hand? 6, because polydactyl is not the most common, but it is dominant. A type of dwarfism is dominant. Huntington's disease is dominant. So let me show you 1 or 2 more pictures really quick of polydactyl.
So you kind of have to have, like, obviously because most people don't have it, have a kid that has it unless you, like, actively...
You cut it off, yeah. I've had 2 students with polydactyl. 2 students with polydactyl. And I'll never forget is my first year teaching AP Bio, and this girl named Samantha raises her hand. It's like, I don't know, this time of year. She'd been in class all year, and she's like, I'm polydactyl. And I'm like, don't look at her hands. Don't look at her hands. Don't look at her hands. Keep looking at her face. Don't be weird. And she's like, you can look at my hands. I'm like, okay. Like that, and I'm like, but you only have 5. She's like, well, it was cut off. And I was like, where was it? Usually if you're polydactyl, you have an extra like half digit on your pinky or on your thumb. And hers was on her thumb. And you could see just like a teeny tiny little scar. And after that, I'd watch her like hold a pencil. and it looked like a claw, looked like a lobster claw. Because for me, I was never born polydactyl. So I have an opposable thumb. It's like kind of like hitting at a 90-degree angle. This is going here, and then this is like 90. Her thumb, because she had two thumbs, so to speak, this one was like off-center, and that one was off-center. And they cut that one off. So when she held a pen, instead of it looking like this, hers looked a lot more like, and I can't do it, but that knuckle was like right there, like 180. It was much more like lobster claw-like. And she's like, yeah, I had 6 toes on each foot and everything else. I had another student, same thing. Hers was on her pinky, and she was like 6'6", also. So polydactyl.
Is it more common in certain regions of the world? Or is it more like
The 2 students I've had, one was African American and one was Caucasian, so I'm not sure. It might be. Like if it started in one major gene pool, then it should be more common in that gene pool. Kind of like blue eyes in Ireland, for example, or red hair in Ireland. So it could be. So generally we just cut off the extra digit because otherwise it'll just be hard to buy shoes and people will probably make fun of you. However, sometimes, nothing new in the chat. Sometimes they get a little bit better. Let me switch cameras for you guys. How many toes does that person have? 6. But here's where your skin is. You can kind of see a faint outline. Maybe not if you're at home. That's where your flesh is. So nothing looks weird from the outside, except that they're 6 instead of 5. Like if you're wearing these, if you had like open-toed shoes and I walked by, I'd just be like, doo-doo-doo. I'd be like, what was, everything looks fine, except all of a sudden there's just a 6th one. There's not like some weird thing shooting off the side. So that one is pretty far back. Like the split is like pretty far up there into your foot. This is from a Radiological Society webpage. This is the most impressive one I've ever seen. Also from a Radiological Society, it's like a bunch of hospital doctors sharing information. That 6th finger goes all the way back to the wrist. So this person might, if I've turned on the hedgehog pathway genes that say build a whole finger, I might have turned on the genes that say build a whole muscle that controls it. So I might have an extra tendon in there, an extra muscle in my forearm, and everything. So this person might actually be a 20% better guitar player and piano player. I was saying as a joke earlier that this person might actually have like decent control of that other finger. The baby that we saw at the beginning, that one's just a slave to the pinky, right? That's no different than if I like taped a Expo pen cap to the side of my pinky. Whenever this moves, that moves. But this one right here, he might, I can't tell because it's an x-ray, so I can't see flesh, just the bone. But he might actually have like a muscle attachment and everything. So he might, how do you play that? And it's like, you can't. I have 6 digits. So pretty cool. Last thing I'll say about that is dominant is whoever wins out if there's a tie. Wild type is what's most common in nature. So 5 fingers on each hand is what we call wild type. But 6 fingers on each hand is dominant. So dominant and wild type are different. Dominant is not always the most common. We get into all kinds of interesting things like that in the Mendel unit. But this will be the last time that I see you guys before semester exams and stuff. So we'll put pause on that and then come back in January. Yes?
This is just kind of a fun fact. Do you know who Anne Boleyn is?
Like, is she a student?
No, she's
Okay.
like the first or second wife of like Henry VIII.
Oh.
Elizabeth the first mother. Well, it was believed that she was, is it called polydactyl?
Yeah.
I might be saying that wrong.
Polydactyl, yeah.
Okay, so it was believed that she had like a 6th finger, and so in like almost every portrait ever painted of her, she wears like really long sleeves on her gown, and it's another reason why she was believed to have practiced witchcraft.
Are you kidding? So they think because she had a 6, I guess that sounds right for like back in the day. It's like, you have 6 fingers, you're a witch. Wow. I didn't know that. What was her name again, did you say?
Anne Boleyn.
Anne Boleyn. That's a cool fact. I'll have to look that up. I can probably find a picture of her or something like that. That'll be interesting. Thank you. Oh, yeah, there's no bells, so I think K-Cal's leaving. I don't know exactly. We're supposed to leave at like 04, so maybe we have another minute or so. But cool. Any other questions or comments from you guys on Zoom? I'll turn off my recording part here.